Welcome to my 25th video in my 30-day challenge. In today's video, we're going to be talking about the second erroneous assumption made by scientists doing research in origin of life today. That assumption is that the initiating molecule of life was a self-replicating molecule. I think most believe that that molecule was capable of replicating itself. I don't know why that is such an ideal situation to look for when we have no evidence of that happening at all in modern life. I believe that was a distraction, that someone who was highly respected came up with that idea and everyone ran with it. What they ended up doing was being distracted and looking for molecules that had the capability of replicating itself, or creating looking for a situation where de novo molecules would create RNA, go straight into an RNA molecule. And that, obviously, because of my theory, was not the way to go. I went the other way. I went to see what were the simplest forms of life, going back from multicellular organisms like us, what kept us alive, what kept us going, then working backwards to simple single cellular organisms. And of course, there's a whole zoo of different types of single cellular organisms with all different types of metabolic activities, all different types of genetic expression, genetic translation, plus the self-assembly of viruses also made it abundantly clear to me that life would have evolved further from viruses, because to maintain a cell you need hundreds of different genes. Meanwhile, viruses had the smallest virus that I know of is the TMV, tobacco mosaic virus, only has 4 genes. And it's a very simple self-assembling system and disassembling system as well, because it reacts solely to the pH of the medium it finds itself in. So if the pH is right, the proteins would become rigid and attract each other and attract its RNA component and envelop it and self-assemble. That to me was a very illuminating fact, that if you look at evolution and the direction it goes, that would have been a very limiting structure to expand on. That would have been the end of its ability to develop further, because what else could a TMV virus do to further encapsulate itself and expand its genome? If we look at it from an evolutionary standpoint, if a gene in a tobacco mosaic virus would change, more than likely it would be a detrimental change for the tobacco mosaic virus, where it would create an error in the proteins, and then those proteins will be unable to self-assemble correctly encapsulate the RNA correctly, and therefore that virus would become extinct. That is, again, what we see in 99.9% of all the species that have ever existed. They're extinct. And it's because they did not either adapt quickly enough for the changing environment, or they had detrimental genetic mutations that made their offspring not robust enough to survive in the current environment, or other factors. But the reigning factor over all of life is extinction. There's a lot of systems within the organism that have to be balanced and work correctly, as well as the other environmental factors that have to be stable enough to allow that animal to continue to exist in that niche that it finds itself in. And if any one of those factors change in a detrimental manner, the most likely thing for that organism is extinction. Not adapting, definitely not changing within the lifespan of one organism to be able to adapt to changing conditions in the environment. So, that goes against genetics, and mutation. And the way we know that evolution works is little changes over time to the genetic structure of organisms that make it beneficial to that organism in its environment, or in its reproductive cycle, or in its ability to metabolize. So to think that a molecular construct living on its own would produce a sufficient complex copy of itself to continue evolving on its own, I think it's untenable. The ability of RNA, the catalytic ability of RNA, is only to cleave itself, cleave other molecules, but it's to cleave. We are unable to make it do other types of catalytic processes. And these are in pristine, controlled laboratory environments. For that to happen in the chaotic and messy environment of the prebiotic oceans, or environment, whichever, clay, surface areas, or volcanic vents, wherever you want to put it, is untenable. It's very highly unlikely that RNA would have behaved any different back then. And again, for that to in itself create a self-sustaining system of production of RNA, I also find untenable, because where would these nucleotides come from? Even though it's a self-replicating molecule, if there ever was such a thing, it would need resources, and the only resources it could use is RNA nucleotides. Where would these nucleotides come from? What system would be producing it in sufficient amount and an efficient consistency for these RNA molecules to self-replicate? So there's a lot of things that a self-replicating RNA needs to overcome, not only in its self-replication, but to find itself in an environment that will provide sufficient resources in the form of nucleotides ad novo from inorganic material source that is non-repeatable. You know, the formation of RNA from dehydrating and hydrating pools would only work for a limited amount of time before there was a sufficient accumulation of water to make the oceans. That once that's done and it's covered, you know, where else would the RNA nucleotides come from? Whereas in my theory, being that the oceans are laden with amino acids that are bombarded upon the earth to this day, day and age, 40 tons of organic material fall from space into our atmosphere. That's now. Back then, it was metric megatons. I mean, you know, the heavy bombardment, you had these asteroids and meteorites that were loaded with amino acids, and the little bit of water that they contained, by the sheer number of impacts, was able to create the oceans we have today. So if there was that much meteorite material to bring that amount of water, certainly it had much more amino acids to bring as well. So what we have in life now is amino acids in the form of proteins working on transforming other amino acids into precursors of other molecules that are made by other enzymes, other amino acids, who then again take that transformed amino acid and convert it into another more complex molecule. But always the starting point is amino acids. Life uses amino acids. Life takes other life forms and only breaks it down to its amino acids. It doesn't break it down to carbon. It doesn't break it down into oxygen. It doesn't break it down into iron. What it does is breaks it down into useful amino acids. And any metabolic process that is performed by living cells starts with amino acids, the transformation of amino acid into another molecule. So what researchers needed to do way back then, which is what I did in the early 2000s, is look for environmental conditions that would allow for this process to begin. How would amino acids bond and take the conformation to transform other amino acids into useful molecules? And here's the thing that I wasn't sure that I elaborated sufficiently in previous videos. Genetic expression is solely translating genetic DNA or RNA bases into amino acid sequences to make proteins. Now, the way these proteins are activated or disactivated in the cytoplasm, in the cytosol, whichever you want to call it, the liquid part of the cell, is by the pH of that cytosol. That pH allows that protein to become, one pH would allow that protein to become flexible and non-rigid and allow it to encapsulate another amino acid or molecule. And then once it goes into another part of the cytosol that has a different pH, then it will become rigid, take a conformation, envelop that other molecule, and perform a catalytic event on it. So a lot of processes that life's metabolism work is based on the pH that protein finds itself in. For instance, hemoglobin. Hemoglobin releases oxygen at a certain pH. At another pH, which will find itself in its lungs, it would encapsulate oxygen, grab it. So that's another means of control, how pH can control metabolic activities. Again, I talked previously about ATP, how it's produced. It's produced by a pH gradient and the protons going through a protein, and that protein taking that energy and creating ATP. Again, the differential of pH. So how did all this begin? How did this become the norm for life to work? There's no wires connecting the nucleus to the proteins to tell them when to activate and when to shut down. There's no wires or communication from the nucleus to the cell membrane to tell it how to do its work and when to do its work. These systems are controlled by pH and the balance of homeostasis created within the cell. So the point is that they were distracted by trying to figure out how to make RNA from inorganic material, and then having that RNA be able to self-replicate. Now, first of all, they've been having a hell of a time to get RNA straight from inorganic material. Whereas if you create the conditions of the chaos engine in my theory, which is the currents passing amino acids through different pH levels within the ocean, create systems that will allow not only the amino acids to bond and become elongated, but also to take certain conformations and be able to act on other amino acids, so which would mimic the cytosol within cells. So that is a much better, easier solution than a self-replicating RNA molecule. First of all, having RNA being made from inorganic material is a very hard thing to do. I mean, they're failing at doing it in pristine laboratory conditions. Imagine having that happen in a messy, chaotic environment way back before life started. I hope you found this video informative, enjoyable, and please give it a like, share it, spread the word to your friends about my theory, and if you care to, buy a shirt or my book, and I'll see you next video.